All right. We're going to get started on old ATP, adenosine triphosphate, today. We're going to get into enzymes, and we'll finish enzymes probably Monday. Okay. Tuesday will be student-led review. Basically, you get these out of study time. Wednesday will be an exam, multiple choice, and free response. Sound good? Also, Tuesday, your FRQs are due.
Oh, can I turn those in?
Can you scan them and put them on the document?
Yeah.
Okay, that works, right? Okay, that way, I'm going to put it where everybody can see it. Okay.
Oh,
So, what is ATP?
Adenosine triphosphate.
Okay, what's it do for us?
Isn't it long-term energy storage?
No.
it makes them good.
What's the opposite of long-term energy storage? Short-term energy storage, all right? Glucose would be more the long-term energy storage, and then even longer-term than that would be your kind of your carbohydrates, things like glycogen, starch, things like that for your longer-term storage within the carbohydrates.
Would photosynthesis be short or long?
It makes glucose, which would be technically going to get like really in depth. Like glucose is medium, carbohydrates like starch and glycogen are long. And they make each.
Well, how do plants get starch and stuff?
Okay. Photosynthesis makes glucose. Plants have processes within them to basically link those glucoses together to make polymers called starch, which are carbohydrates. Does that answer that question? Okay. Because you remember those carbohydrates, it's just simple sugars stuck right next to each other. Basically, the monomer is going to be a monosaccharide, usually glucose, and the polymer is multiple glucoses, sometimes fructoses, stuck together for a long chain. All right, adenosine triphosphate. In this section, we're going to learn what provides energy for cells, inorganic reaction. It's usually the hydrolysis of ATP. If you remember, we talked about inorganic, it absorbs energy, so you have to add energy no matter what. Exergonic, it will release energy, and those will happen spontaneous, meaning you don't have to add a bunch of extra energy. You still need to have enough to reach that activation energy, but usually it is going to be just the ambient heat is going to be enough to do that. So when delta G is greater than 0, what kind of reaction is it? Inorganic. If it's less than 0, it is exergonic. What is delta G? Change in free energy. Very good, right? Okay. Remember, endothermic, inorganic, relatively the same thing, except we're talking about energy instead of heat. Exergonic, exothermic, kind of the same thing. So ATP is composed of an adenosine backbone with 3 phosphate groups attached. This is actually just a modified nucleotide. Nucleotide being the monomer for nucleic acids. So you're going to have a ribose sugar. You're going to have adenine as your nitrogenous base. And then you're going to have 3 phosphate groups. You remember from your CK12 stuff, a nucleotide is going to have 1 phosphate group, 1 sugar, and 1 nitrogenous base. Right? So essentially what we're doing, we're adding 2 phosphate groups onto a nucleotide. And then the energy storage and transfer happens with this last one. It's called I think the gamma phosphate. Yes, the gamma phosphate.
What is this section called?
ATP. Adenosine triphosphate. Kind of check in. You still liking the way the class is set up?
Yeah.
Okay.
I like the reading checks.
Yeah.
You know what I do too much.
I just messed up.
Yeah. Again, this is just another visual of ATP. You've got your ribose sugar, adenine, your nitrogenous base, your 3 phosphates, alpha, beta, gamma. Don't know the Greek alphabet yet? Alpha, beta, gamma. Alpha, beta, gamma, delta, beta, iota, gamma, suicide, combine. When I rush for a frat, you have to say the whole thing before a match touches your finger. I know I didn't rush a frat. I'm just a nerd that knows how to do it. Okay?
Do you actually do that though?
I've heard, I don't know for sure. Okay. But
Try it out first, Mr. Rowell.
Say what?
Just try it out first.
No. I'm good. There'll be no, you just want to see me get burned.
Maybe.
Maybe?
Not.
Yeah.
It's a challenge.
Challenge. Alright, so the structure. The nucleotide consisting of a nitrogenous base, adenine. Do you know what the other 3 nitrogenous bases are, or 4 nitrogenous bases?
Adenine, thymine,
Glycogen.
Guanine,
and
cytosine.
cytosine.
Cytosol. One more.
Uracil.
Uracil, right? Uracil is an RNA. Speaking of RNA, is ATP closer related to DNA or RNA?
DNA.
Why?
Because it is.
Is it RNA transport?
Because, dang it, I knew this question.
Oh yeah, right.
Dang it.
RNA is ribonucleic acid, DNA is deoxyribonucleic acid. So this is closer in structure to RNA. Yes, sir. I have been. I need a good friend.
The one by Goodwill. The one behind Goodwill.
Alright. So the three phosphate groups, in order of closest to farthest away, alpha, beta, and gamma. The bonds that link the phosphate groups are high energy bonds. When the bonds are broken, the products have lower free energy than the reactants. Alright? Because the products have lower free energy than the reactants, is this going to be an endergonic or exergonic? Exergon, products releasing energy, your products or your reactants have lower energy. Good job. Golly, I'm just hitting on that structure all day, aren't I?
Mhm.
Jeez, that's right. That means you should definitely know it.
So Mr. Rowell,
Yes, sir.
are you gonna post the slides themselves or just the?
I'll post the slides, yeah.
Okay.
But Zach, I'm not done with this section slides, and so once I do that, I'll post it.
So you finish the chapter.
Yeah, seriously.
Would you say that the structure is just adenine, triphosphate, and then ribose? Or do you want to be strong?
No, that's good. You got a nitrogenous base adenine, you've got a sugar ribose, and you've got 3 phosphate. Yes, ma'am.
You think, are we gonna have like time to do our food response after this lesson? Or?
Well, probably not. We're going to get as far as enzymes as we can. Okay. Usually, most weeks, it's going to be pre-response time is going to be outside of class.
Oh.
Okay. Right. That's why I give you like a whole over a week to do it, I think, or about a week. Right. So I recommend getting like a Google Doc you can share, divvy up the work, so on and so forth. Right. Okay. All right. So the hydrolysis of ATP. Hydrolysis. Y'all remember what that means?
Water.
Oh wait, it's
Hydro means.
water.
Lysis means.
Process.
No. Philic means. Say what?
It's like a process.
No.
Break stuff down.
Break stuff apart.
Yeah.
Very good, right? So hydrolysis is just breaking apart of water, and this also breaks apart molecules, okay? So AT plus H2O yields ADP plus PI. That's free energy. The delta G for this is going to be negative 7.3. So what type of reaction is it? Exergonic, yep. It's an unstable molecule, so it hydrolyzes quickly. If it's not coupled with an endergonic reaction, this energy is lost as heat. So essentially, this loss in energy is usually transferred to some other type of reaction. Usually, it attaches to a protein and makes it move.
Do we need to know that?
What?
Okay.
No, you need to know that hydrolysis is the way ATP gets broken down into ADP.
ATP.
This is important. ATP hydrolysis is reversible. Do you know what the mechanism of that reversibility is? Cellular respiration. You got it. Okay. So that's a process of making ATP. That's pretty much its only job is to make ATP. What does that look for, Mr. Harrigan?
Just trying to remember. ATP hydrolysis is
I'm trying to tell you. Well, I listened to that YouTube recording, and it feels like I talk so slow. Do I?
No.
No.
Okay. Because I'm listening, I'm like, jeez. If I was listening, I'd fall
have the
asleep.
playback speed slower.
You slow it down. I remember I used to watch some television shows, and I'd speed it up like 1.5, 1.75. I'm trying to read notes.
I swear your brain's working quicker than it.
Yeah, I just see
So many different levels.
Hey, someone did beat you. Last year I gave a quiz in physical science. It was 10 questions, multiple choice. It was when I was gone.
Oh yeah, I remember that.
One kid got them all wrong. I was like, geez, how does this happen?
We had to have known them all.
I don't know.
I
Wasn't it Brendan?
I actually tried.
No, I wasn't Brendan.
I guess that's very important.
These ones are
It's
thin.
Yeah.
probably like every other
I do
slide.
want you to make sure to know it. So when we talk about the bond that gets cleaved, it's this one between the beta and the gamma phosphate groups. So this is where it gets bond. Or it gets cleaved, right? So just kind of a reminder, there's dehydration synthesis and hydrolysis. Dehydration synthesis, you basically put stuff together and then you make water. Hydrolysis, you break apart water and then you break apart molecules. Okay? Catabolic or anabolic?
No, antibiotic.
Anabolic.
Anabolic.
Not this one.
Catabolic.
Catabolic, right.
Because cats break things.
Yeah, cats break things.
Anakin built the Death Star.
Anakin built the Death Star. Good job. This is just another visual. So we've got ATP, it's the high energy molecule. ADP, adenosine diphosphate. Di stands for 2, it has 2 phosphates. So it is, the lower energy molecule. And again, you've got hydrolysis, you break apart water, and you need ADP plus energy in the phosphate.
I could never imagine doing coffee.
What?
So I could never imagine doing coffee or caffeine drinks.
Well, I've got one right now.
Like, I mean,
Oh, you want to hear me talk fast? Is that what you're saying?
Yes.
Yeah, like
Yes.
I can talk.
The first
I don't
day
know.
of last.
Did I do that on purpose or?
I don't know.
Oh no, that's just my natural speed.
Yeah.
So I have slowed down a lot. When I gave presentations, that was always the joke, is that I go super fast. Yeah.
I think we got through that first.
Can you be an
one.
auction
It took
person
5
as well?
minutes.
Say what?
Will the auction person?
No, no.
Country
auctioneer.
auctioneer.
No. But that was always the joke, is like I always go fast, and that was always my critique, is you gotta slow down, you gotta slow down, you gotta slow down. You gotta slow down and you gotta stop moving so much.
You still move a lot.
Mr.Rowell.
So what?
You still move a lot.
Yeah, I do. And it's strategic.
I don't
It's
know.
like, would it be a lot more, I figure it would be a lot more boring if I just stood.
Yeah.
Yeah, like this,
Yeah.
like when Mr. does that, it's like
It's.
Lois Phillips, she like bounces around the room and
Who
dances.
does that?
Mr. Rowell.
Sir.
When you retire, you should become a auctioneer.
No, I'm good.
That'd be crazy.
I'm gonna camp and hike when I
Oh, no,
Come on
no.
now. Alright, the sodium potassium pump. It's an example of energy coupling. So the energy derived from the exergonic ATP hydrolysis is used by the integral protein to pump 3 sodiums out of a cell and 2 potassiums into the cell. Because we're using ATP, what type of transport is this?
I mean, active.
Active, right? The energy is coming from ATP. So in general, when we see stuff in the body, we will pair endergonic and exergonic reactions, right? So since this is exergonic, it's releasing energy, the reaction of this will be what?
Active.
Endergonic, right? So this reaction's losing energy, this one's gaining it, okay? And it's using this energy to push stuff against the
Concentration gradient.
concentration gradient, right? That's active transport. There we go. Uses ATP and it's against the concentration gradient. Alright, the previous illustration depicts the sodium potassium pump, which uses energy derived from ATP hydrolysis. The hydrolysis of one ATP molecule releases 7.3 kilocalories per mole of energy. Delta G equals negative 7.3. It takes 2.1 kilocalories of energy to move 1 sodium across the membrane.
Chase.
Give me 1 second. Delta G at plus 2.1. What is the maximum number of sodium ions that can be moved by hydrolysis of 1 molecule?
Give me 3.
Who thinks it's A? 5.
4.
Who thinks it's B?
Oh yeah.
Who thinks it's C?
Give me
Oh,
3.
yeah.
Okay, thanks 2. It is 3.
Oh
Yeah, yesterday on our reading quiz thing, it says wrong, but we all put 3 on it.
Yeah, I think I corrected it.
Okay.
You did.
Oh, is that what you asked
I'm
me?
at 100
Is
on
what
that
the.
Yeah, because essentially if it takes 2.1, how many times did 2.1 go into 7.3? 3 times. Right?
You can just
Yeah.
The hydrolysis of 1 ATP releases 7.3 kilocalories of energy. It takes 2.1 kilocalories per sodium ion. And again, it'll get 3 across.
Here, that's
Alright, so section questions. Why is ATP considered the energy currency of the cell?
Because isn't it like
It's
It's
powers
made by
a lot of
bottom
that.
line
common.
Transport.
Say what?
It powers all of that.
It powers a lot of the active transport. It's also going to power anything within the cell that needs energy. Okay? Anything that needs energy in some way, shape, or form. Now, it could be powered directly, either by ATP attachment, or do you all remember that secondary active transport?
Yeah.
Okay. We use ATP to build
um
to do active transport to build a
gradient?
concentration gradient, and then we get to use that gradient we've made using ATP to power other things, right? It's kind of like a water behind a dam, right? Make sense?
Yeah.
So pretty much any reactions or some way, shape that happens within a cell, powered by ATP. How is energy released through the hydrolysis of ATP? Hydrolysis does what?
breaks down.
Breaks water, breaks stuff apart. What happens when that gamma phosphate is removed? It does what?
Say kinetic energy, that's wrong.
I mean, energy. What does it do with energy? It releases it. Okay, and it's usually, so if it releases energy, is it endergonic or exergonic? Exergonic. Usually in the body we pair that exergonic reaction with what? With what? No. We pair an exergonic reaction, an energy that releases reaction with an endergonic reaction. Okay? So that things can get done. And that the example that was given here again, started potassium. This is an exergonic reaction, it releases energy. This is an endergonic reaction, it takes energy. So since we take energy from here, it's lost, and then we put it into here so that work can get done. Yes, sir. Inorganic phosphate. Okay, does that answer your question? Okay. So this PI, so we've got ATP here, ADP plus a phosphate group. That's your inorganic PI. Okay, so it's essentially this is gamma phosphate after it gets cleaved off.
Oh,
Okay, does that make sense?
so like when ATP turns into ADP, it gets cleaved off, and that's your pot?
Yeah, that's your phosphate. So this is the third phosphate. Okay.
Okay, so it's just floating around now?
Yeah.
Okay,
Yes, sir.
got it.
All right, thank you.
Wait, so endergonic releases.
All right, any questions on ATP?
Yes.
Yes.
Endergonic is the releasing of energy?
No.
It's the breaking down.
Endergonic, you have to absorb energy.
No, absorbs exergonic.
Releases.
Okay.
Yeah. Anything else? So y'all know the structure of ATP.
Yes.
Y'all know the reaction of ATP to release energy.
Hydrolysis.
Hydrolysis. You go from ATP to what are the resulting molecules?
ADP.
ADP and a phosphate, right? That releases energy. That energy is used to power other stuff in the cell. For example, active transport. Yes, ma'am.
So the hydrolysis does what?
It releases the energy, right? It breaks off that third phosphate, that gamma phosphate, and it releases energy.
Okay.
Hydrolysis means breaking apart water. You have to split one molecule to do that as well.
Okay.
All right, cool. 6.5 enzymes. We're going to go through this as much as we can today. I'm going to be honest with you, this presentation's not the best. I'm still only halfway done working on it. So if we run into where I stopped, we're going to stop too.
Oh, yeah.
Got it.
We got 20 more minutes.
Yeah, we got 20 minutes. 29, okay.
Don't let them know
On
that.
my watch. That was a minute slow.
No, ours. I'm looking at mine.
Yours is fast. No, yours is, I don't know.
Yours is
All right, enzymes. These are protein catalysts. Okay? Meaning, what are they going to be made of? Amino acids. Very good. We're going to hit on protein structure again when we go throughout the section. So these will bind with reactant molecules. So when we talk about enzymes, the reactant molecules will be called substrates. Promoting either bond breaking or bond forming, depending on if it's an anabolic or catabolic reaction. The enzymes are very specific. They will only catalyze one reaction. So they're not generalists, they're specialists. There's one reaction that they will catalyze and nothing else. Okay.
In a
So-
lifetime or at once?
Period. In the lifetime, right? If you have an enzyme called lactase, which breaks down lactose, that's the only thing it does.
So, with lactose intolerant, do they just not have enough of those enzymes?
Yeah, we're actually going to talk a little bit about that. Okay. Because you can actually take a supplement Mr. Rowell knows.
Do you take it or no?
To help you digest lactose, okay? Because lactose, it's a disaccharide of two different sugars. I'm not sure if it's like two glucoses or two fructoses, I can't remember. Or it's like a glucose and maltose or something.
Actually, y'all not being lactose intolerant is not.
I'm like slightly.
So, while the overwhelming majority of biological reactions that happen in your body are the result of enzymes, there's kind of like a subclass of RNA molecules called ribozymes. They can also catalyze reactions. But the vast majority of them, 95+%, are catalyzed by enzymes. Any questions on that? Yes, no, maybe, kind of, sort of.
So, this might be stupid.
No, go ahead.
There's only one type of enzyme?
No, there's thousands of types of enzymes.
Okay,
It's-
so
Go ahead.
each enzyme catalyzes one thing?
One type of reaction.
Okay.
So think about all the reactions that happen in your body, everything from being able to break down food to touch your nose to fight off infections, anything like that, right? There's a specific enzyme for each and every single one of those reactions. Each enzyme or type of enzyme only does one type of reaction.
But do we have multiple of those?
Yes. Yeah, we've got probably hundreds of thousands of enzymes. sometimes probably within the same cell. So,
So are like white blood cells just like a pure version of the enzymes that attack?
no, white blood cells are cells, and so they're going to have all the same components of the cell. You know, nucleus, mitochondria, cytoskeleton, all that jazz. But some white blood cells have additional enzymes meant for breaking down more nuclears. But those will be specific to breaking down basically a carbon-carbon bond that might be an invader, a carbon-hydrogen bond, or whatnot. They're specific to breaking those type of bonds, if that makes sense. All right, so the enzymes basically lower the energy needed for a reaction to occur. Usually these are going to be endergonic reactions. However, sometimes you can also catalyze exergonic reactions as well. Yeah, it lowers the activation energy. We'll get to that in a little bit. These are some examples of enzymes. Lactase breaks down lactose. You're going to find this in your small intestine. Amylase breaks down starch. Pepsin breaks down proteins to amino acids. You're going to find this in your stomach. Lipase breaks down fats to fatty acids. And then alcohol dehydrogenase breaks down alcohol to non-populated substances. This is where you're going to get into the early stomach. Yes, ma'am.
So enzymes are catalysts,
Yes.
but I thought catalysts sped up.
They do.
But why does it lower?
What do you mean? It lowers the energy needed for the reaction to occur. So think about if a reaction is going up a hill and then getting to once you get up that hill, then you can roll down. What enzymes do is they lower the hill. So you don't need as much energy to get to the top of the hill before you start rolling down. We'll see some figures as we go throughout this and make it a little bit more clear. Does that make sense?
Yeah.
Does it really, or you're just gonna be like, I'm just
No,
gonna
like,
say
yeah.
Yeah, the
Catalyst, I was kind of confused there for a second, but it honestly.
Mr. Rowell, can I take my room or bathroom basketball?
There's a huge line.
Go ahead and go.
Yeah, you can check. I'm not going to.
Yeah.
What are all these enzymes? Most of them end with
Ace.
ACE, right? So when you hear the word ACE, or you hear that ending, think enzyme. And usually that's going to be breaking down something. All right, so this is kind of what Ms. Thompson was talking about. So this is a reaction, endergonic or exergonic? Endergonic, right? It absorbs energy. Products have more energy than reactants. So the purple line is going to be your activation energy without an enzyme. The red line is with the enzyme. As you can see, it lowers the activation energy, so you don't have to push as high up that hill.
Have you read your disc?
Yeah, I'll take a picture. Okay. So how does bond an enzyme? And each enzyme is specific for a substrate. Substrate is just another way of saying a reactant. Most enzymes end in the letters ASE, ACE. So the enzyme that breaks down sucrose would be
Sucrase.
sucrase. Yeah.
Oh, yeah.
Okay. The enzyme that breaks down lactose is lactase. Very good. Okay. Now you can actually buy some enzymes. So Mr. Brown might know about these. Yeah. So if you are lactose intolerant, if you don't have that enzyme to break down that milk, you can do this, right? Helps prevent gas, diarrhea, bloating.
Oh, that's
Hey,
weird.
drop something.
Can I have it now?
No, it's still good. All it did was agitate it and release the CO2. Right? It'll reach equilibrium again.
In a few hours.
Yeah. There's that one.
You can
just say
have
it
it.
extra bubbly. Scientific.
Like the liquid top of it.
I'm good, right? So this is what lactase does. So lactose is basically 2 glucose. No, galactose and lactose. Okay. Yep. It's a giant
It's a
lactose.
Giganosaurus.
Right. So lactose basically helps break the bond between the glucose and galactose, and then the body can absorb these sugars.
Crazy.
All right, properties of enzymes. One, they are made of proteins. So if you hear enzyme, think protein. They speed up reactions, and they remain unchanged after each reaction. That's part of the definition of a catalyst. The catalyst is going to be present at the beginning of the reaction and at the end of the reaction. It'll be the same at the beginning as it is at the end. It's not changed as part of that reaction. That means it can be reused indefinitely until that enzyme breaks down.
That thing looks like that one little figure of the nerds. The purple
Yeah.
Yes.
So let's say we've got this enzyme sucrase. The site where the reaction happens that it catalyzes is called the active site. So the substrate will come in, sucrose. Sucrose is a disaccharide. It's I think it's a glucose and a fructose put together. The substrate will bind to the active site, creating what is called an enzyme substrate complex. There's no substrate, it's another word for reactant. It will convert it to its products. Here we have water coming in, so what kind of reaction do you think this is going to be?
Hydrolysis.
Hydrolysis, right? We're adding water to it, and we're going to break it apart, and we release glucose and fructose, and again, that enzyme can be reused at that beginning of the reaction again. It can keep on going, keep on going, keep on going. And going and going, like the Energizer Bunny, right?
I'm sorry, that thing's never stopped.
Alright. You've probably heard me say this multiple times. You're going to hear me say it the rest of the semester and year. Structure drives function. So when we talk about the 3D shape of the enzyme and reactants, determines the specificity. Okay. Some people, when they talk about enzymes, it's a lock and key mechanism. How many keys fit within one type of lock?
Structure drives what?
One structure drives function. One type of key. Same with enzymes, right? Only a certain set of substrates or reactants fit with that enzyme. Substrate molecules interact with the enzyme at the active site. That's where that reaction occurs. Enzymes can catalyze a variety of reactions. In some cases, two substrates bond together to form a larger molecule. In others, one molecule will be broken down into smaller products. So it can be catabolic and anabolic reactions.
So enzymes can do both?
Yep, enzymes can do both. Yes, ma'am. Alright. Thank you.
I think it's good.
If I wanted
It's
to make
just
sure
flat.
it was good, what could I have done? You physical science students.
You could just like slowly open it, like really
I don't remember.
So
That's what I did.
CO2 solubility increases as you go down in temperature, so you can go to the fridge. And the CO2 would be absorbed more.
Every day, you knocked over my sparkling water. Can I remember that?
Applied force.
Yes. It was always flat by the end of your class.
Wait, I have a question.
Yes, ma'am.
So if they can do both catabolic and anabolic, you probably already answered this. Hydrolysis or whatever. They do that?
So yeah, they can do hydrolysis and they also do dehydration synthesis, which is the opposite of hydrolysis. Yes, sir.
So, like anabolic steroids, why are they called anabolic?
Probably because they help the muscle.
Okay, I didn't
Don't they stop your growth?
What you mean, Gary? You take it some?
know.
I don't know about
That's what he is in science.
He asked that question.
I know that like
No, he's thinking left.
a lot of protein.
Wait, you're thinking.
They what?
I thought they gave you like a lot of protein.
They'll give you a lot of proteins. It might help you metabolize and build more proteins.
You know exactly what
And
I'm
there was like more.
I'll let
Because you can't just make protein, you have to do proteins.
I saw on TikTok, it was like anabolic, and I'm like, we just went over that.
It probably helps build the proteins within your muscles better, is my guess. I don't know.
I got you. They know to give drugs to testosterone. They are used to increase muscle size, strength, and performance. So it's like a testosterone.
Yeah, it builds muscle. That's what it does. Alright, so enzyme substrate specificity. This example, we have a substrate. What is this thing called? Active site. Do y'all want to do it on that?
Yeah.
That one or this one?
Both.
Alright. So I'm trying to mind. This is called the enzyme substrate complex. Then the reaction will occur, the products will release, leaving the active site of the enzyme open for another reaction.
Oh, that's scary.
Alright, so this is kind of a three-dimensional figure of an enzyme. As you can see here, this is the molecule that's catalyzing. The blue area will be the binding sites, and then the red area is what's called the catalytic sites. Red area is where it actually lowers the activation energy. So this is where it kind of sticks. This is where it helps to lower the activation energy. At the active site, there's a mild shift in shape that optimizes a reaction. This is called induced fit. So the slight changes at the active site maximizes the catalyst. The induced fit is a relatively recent discovery. It's used in expansion on the previously held lock and key model. So let's say that we have a pencil. Yeah, we'll do a pencil, right? So what induced fit does, especially if you're doing like a catabolic reaction, the enzyme will come in and it'll put stress on the molecule so that you don't have to add as much energy to break it, okay? So if you're doing breaking stuff apart, it kind of puts a little bit of stress on it. Since that enzyme is putting the stress on it, you don't need as much energy to break it. Now, if you're doing a dehydration synthesis, essentially what happens there is the enzyme will help orient it. So instead of these floating around and expecting them to come together perfectly within the cell, the enzyme will basically be like, all right, I'm going to bind to this, I'm going to bind to this, and then I'm going to put them back together. Okay. Basically, it just orients it in the correct fashion so they can be stuck together and bonded
Oh wait, they're standing.
Okay.
That's a waste of patience.
You're a waste of a pencil. No, I'm just kidding.
Man,
So basically, the catalyst
that kind of hurts.
just
Just
does the
leave.
It lowers the energy needed to
Huh? What?
makes it easier
What?
for it to happen.
What?
The size of an eraser or something. You got one.
That was quite a tender, wasn't it?
Yeah, just that then you spit on it again,
It's okay.
so on and so forth.
I don't want.
Over here, you've got the catabolic reaction. Substrate will come in, bind, be split across, you have an enzyme release. Here's going to be a anabolic reaction. Substrate will come and bind, stick together, and then releases as well. Yes, sir.
Can you give me those pictures? Why? So I can move forward. I understand where you are.
You won't
You won't have to.
have to.
Yeah, I'll have the option.
This is kind of a three-dimensional view of that induced fit model. So you got yours. What are these called?
Enzyme.
This is your enzyme.
Right.
Nope.
Doodles.
Substrate.
Doodles. Woo.
Substrate. Substrate of the reactants in an enzyme reaction.
That's doodle.
What is this area called?
That's your
Activation.
Active site. Very good.
I can't see.
What is this whole thing called together?
Protein.
No.
Enzyme. Enzyme what?
Substrate.
Enzyme
It's just like
substrate
that.
what?
It's gonna be like that.
Structure. I don't know.
Complex.
Enzyme substrate complex.
I was close.
You just say random words now.
Is this an anabolic or catabolic reaction?
Anabolic.
Anabolic, right? Yeah, it's putting them together.
Builds.
So you can see how when it first comes in, these aren't exactly that shape, but once they bind to it, the enzyme orients it around it, so it kind of snaps together. Alright, phosphofructokinase
Huh?
deficiency is a genetic disorder that occurs when a person lacks PFK, an enzyme needed to perform glycolysis in skeletal muscles. Applying your understanding of both enzyme function and glycolysis to predict the effect that PFK deficiency would have on the body. Would be the production of energy by glycolysis will occur more slowly than normal. Skeletal muscles will function properly. Production of energy by glycolysis will not occur. Skeletal muscles will function properly. Production of energy by glycolysis will occur more erratically than normal.
Okay, I'm
Skeletal
not
muscles will not function properly. Or production of energy by glycolysis will not occur. Skeletal muscles will not function properly.
Oh, it's one of the last two. Or skeletal. Wait, never mind. They're also skeletal muscles. Wouldn't it be more slow?
Mr.
Think it's 4? Alright, who thinks it's 1? Okay. 3. Who thinks it's 2? What's up? Who thinks it's 3? Who thinks it's 4? So you're right in that it's one of these two.
Who's right?
Mr. Paul, when he said it's one of the bottom two. The skeletal muscles will not function properly. This is going to depend on the enzyme needed to perform glycolysis. Okay, yeah. It'll be this one.
Yeah.
Because if the enzyme is needed to perform glycolysis, can you perform glycolysis without something you need?
No.
So it's going to be this one.
Ask for.
It's cold in sixth grade, Mr. Rowell.
Alright, enzymes are proteins.
Yes.
So proteins are formed from the polymerization of what? Amino acids. It's a peptide bond. What type of reaction is this?
Anabolic. No,
Oh,
catabolic.
no.
Yeah, dehydration synthesis, which would be anabolic, right?
Yeah.
Okay. What are these things here?
Molecules.
Protein.
No, they're
Chemical
no,
structure.
it's
You
under
said it
A's.
earlier.
I mean,
They're amino acids, right? So amino acids going to have a carboxyl group, a nitrogen group, a hydrogen, and these are called your R groups. So I think there's probably hundreds of different amino acids. I think there's 22 naturally occurring with your body. Basically, these R groups are what make each amino acid different.
in and I.
Yes, most of them will. Cysteine, or this one doesn't, aspartate, glutamine, lysine, alanine.
What are those again?
These?
Amino acids.
They're amino acids. The shaded things are called the R group. That's what differentiates one amino acid from another. Okay, the 3D shape of the protein is determined by the amino acid sequence of the polypeptide. That's primary protein structure. The amino acids of the active site are super important to the enzyme's function because they allow the binding with unique substrates. Okay. When we look at these amino acids, the things that do the interaction are going to be these R groups with whatever the substrate is. Cellular binding is also important for function. Suboptimal temperatures can denature the enzyme. Denature is just a fancy way of saying breaking down or losing shape. The enzyme loses shape. If an enzyme loses shape, we talked about structure drives function. Does it still work?
No.
Either not as well or not at all, right? Depending on how odd that shape is. Also, pH can affect that shape too and denature it as well.
How many more slides we got? So,
I don't know. Let me look.
Don't forget to stay.
it does.
Yeah.
Okay, a lot.
Stop. I'm trying to figure out how to.
Escape.
Yeah. Without pressing escape. 179.
Okay, a lot.
Yeah, we have a lot.
You said
Like, which enzymes are going to take 2 days.
Oh, okay.
Yeah, right. I thought I did say we were going to finish enzymes Monday, did I not?
Yeah, you did. You said finish on Monday, but
We said you didn't quite finish the slides yet.
so we thought you got like 5
Yeah.
slides then. And just
Where are we at?
97.
Thank you. So we've got like 35 more slides.
So there's no vocabulary on the test Wednesday?
There's no like matching. It's going to be questions, but you'll have to know the vocabulary. Does that make sense?
So I think I can get through 35 slides in 9 minutes.
No, absolutely not.
No.
100% no.
How many have we been through so far?
Probably 30.
200, huh?
The whole chapter's got like 130 odd slides.
So denature means break apart? Or what does that mean? Break down?
It loses its shape and it breaks down. It's kind of loses its shape. Alright, so these are kind of the variation in those amino acid R groups. That's what makes the different amino acids different. As you can see, they're kind of grouped here. You've got non-polar ones. What do you think? Where do you think these are going to interact with on a cell?
Inside.
Inside the
Phosphate. No, yeah, phosphate? Or the
inside the cell membrane, right? We've got those non-polar tails. What do you think the polar tails are going to interact with? Or the polar amino acids? They're going to interact with water. Water likes polar stuff. Positively charged and negatively charged, they will react with water as well. Keep in mind, what happens in chemistry if we have positive charge and negative charge? Yeah, they're attracted to each other. If we have like charges, what do they do? They repel. Okay. And you've got non-polar over here. When you hear the word aromatic, that means a ring, specifically something called a benzene ring. The aliphatic means a chain. That's an organic chemistry tip for you. All right, kind of recapping back to protein structure. There are 4 levels of protein structure. We've got primary, which is the order of amino acids, or the sequence of amino acids. We've got secondary, which is the local folding. We're either going to get alpha helices or beta helice sheets. Tertiary protein structure is a 3-dimensional folding structure due to the interaction of those R groups. And then you've got quaternary. That's where you've got a protein consisting of more than one amino acid chain. Watch this little GIF. We're going to have to wait for it to cycle back to primary structure, huh?
Yeah.
It's primary structure, order of amino acids, secondary structure, alpha helices, and beta pleated sheets. Tertiary structure is the R group interactions making 3-dimensional shape. Quaternary structure is proteins with more than one polypeptide subunit, which is that amino acid chain.
You're really glad you did that.
I do. They're very good. Because here's the thing, if I show a video, kids stop paying attention by 20 seconds in.
Yeah.
You're like, yeah, we do. Well, yeah, we do. Yes, sir.
Unless it's a song.
Yeah, that.
Mr. Rowell.
Tertiary.
Yeah, that. Most commonly found in.
The
look like a
Mr.
word.
Rowell, unless you can find some songs, I'm not gonna face you.
Oh, tertiary.
Like,
Tertiary.
Tertiary, yeah.
like the song.
What is it
There's
mostly
something
complex?
Like, it was like the fox.
What do you mean, tertiary?
Yeah.
Tertiary just means third.
I know, like that structure. Like, what do you see it?
Tertiary is the
It's
third
a worm.
structure. There's the first structure, second structure,
The
third
gift is
structure,
a verb.
fourth structure.
Oh.
What's
Yeah.
your
Yeah, it's
It's fancy
It's not
way
a
to say.
Look, it's
Yeah.
a
I'm thinking-
quaternary is a fancy way of saying fourth. Alright, again, enzymes are proteins. Lactase is made up of 4 subunits. Each has 1,023 amino acids for a total of 4,092.
Well, that's
So when we talk about the complexity of these protein structures, these enzyme structures, this is kind of what we're saying.
That's
Who
scary.
counted them that perfectly?
That's not
Or what if it's 1,024?
Or 1,022. Wait, take a picture of.
Yeah, I'm taking a picture of Mr. Jones sleeping.
I'm not. I got a confession.
I think it's
Check it
good.
out, that goes.
Yeah, tell.
Let me take that speech.
Hey, Garrett knows something about that. Marcus did.
Alright, so how enzymes work? Enzymes are a protein molecule that functions as an organic catalyst. The substrate, those are the reactants. The energy of activation is the energy required to cause the activation. Again, the substrates will bind to the active site of the enzyme. Bonds in the substrate are weakened in this enzyme substrate complex, allowing it to break off and become the products. The enzyme can help the substrate reach its transition state in one of the following ways. I can position 2 substrates so they align perfectly for the reaction. I think about them orienting the pencil where it'll stick together. It can provide an optimal environment, i.e. acidic or polar, within the active site for the reaction. Those side chains can be either more acidic, less acidic, more polar, less polar to drive that reaction. It can contort or stress the substrate so it's less stable and more likely to react, like bending the pencil so you don't have to add as much energy in it to get it to break. And it can temporarily react with the substrate, chemically change it, making the substrate less stable and more likely to react. So essentially the substrate will react with the enzyme, and then another substrate will react with the enzyme substrate complex, basically providing like a transition rate. So again, this is effect on the activation energy. With the enzyme, you lower the activation energies. When you lower the activation energy, these reactions can occur more quickly. Enzymes lower the activation energy of your reaction. This is showing that visual again, the difference in the activation energy without the enzyme as well within the enzyme. Now, the change in free energy does not change. What that mean? Yes, sir.
So, with different enzymes, does it change the activation energy requirement by the one? Or is it like pretty much the same for all of them?
It'll depend on the enzyme. Because some reactions are going to have higher activation energies regardless. And so not all reactions will have the same activation energy, so the enzyme is going to have different varying effects on activation. Does that make sense? Yeah. Okay. Enzymes do not change the total free energy change. So the energy of your reactants will still be the same, the energy of your products will still be the same. It just changes the path to get there. Am I losing y'all?
I'm just tired.
It's Friday. Tell you what, I'll be nice. I'll give you the last 2 minutes. Tell you that.
Alright.
Woo! It's a dinosaur.
We're gonna
It's
get
not
like
like a
it.
thing from Jurassic Park. Anybody seen that one?
Are we ever gonna get bigger than
Yes.
So essentially, the reason we do small blocks is that's the way that college board has it set up.
You look like a
And
dinosaur.
if for whatever reason,
Wait.
You go this way. You go that way.
Oh, is it like? If I did like the intro, deal. I don't think I can.
And most of them, because you're going to do a lot of files and some of them are
Yeah, that's exactly what's about.
What's up?
I get push control.
Yes.
Someone just hand it to me.
I tell you what, I'll post the ones that I have today. I'm definitely not giving that advice.
Alright. What? You know, you
Good morning, Blake. Yes, sir. Say what?
I'm not your consent.